everybody. Maybe I could just start by thanking Sandra and Christian, who's all the way at the back there, and GRIN Italia for a fantastic conference here. The location's fantastic. The science has been great. It's been great actually to see some of the clinicians and meet with some of the caregivers and patients. And I went to the first meeting. My first time I went to the meeting was in Barcelona, actually, where I met Sandra and Christian. I had a great time there. And because I often teach at this time of the year, I couldn't make the one last year, but I thought I'm going to come here and kind of present our work. So if anybody knows our work, our interest is really, I'm really a fundamental scientist. You'll see in a minute, I'm actually doing translational work as well. But the interest in our lab is really we're sort of interested in the sort of building blocks of the nervous system. So that's the kind of fundamental approach that we take. And that's why I've come up with this kind of rather philosophical title, but I'm going to get into the kind of details of that in a minute. If anybody knows the work that we do, then we spend quite a lot of time looking at different kinds of ion channels, which are found in synapses, so voltage-gated ion channels, which might not mean much for people who are not scientists, ligand-gated ion channels. But one of the building blocks that we really spend a lot of time looking at is the glutamatergic synapses. And so I want to tell you a little bit about that. I want to really kind of build upon what Ian talked about this morning. So I think he and I are the only ones who are really going to be talking about AMPA receptors. So I'm interested in the NMD receptors, so all the GRIN work that's been done, I'm very interested in that. But we're actually also very, very interested in AMPA receptors, so the GRIA genes. And what you might think from that is that the AMPA receptors tend to have been pushed to the sort of uninteresting brother of the glutamatergic synapse. And I'm going to tell you that I think that's probably not true. There's something much, much more interesting that we've sort of been uncovering over the years. And so what I wanted to do is basically take papers that sort of span 10, 15 years from our lab. And although if you read the individual papers, you won't see the story that I'm going to tell you. But if you read very carefully, you can actually see we've been actually unmasking this kind of story that we're almost getting to the end of. We haven't published all the stuff yet on it, but we're always coming to the end of it. It means that the way we think about AMPA receptors and we think about how we develop drugs to AMPA receptors, particularly in the context of GRIN disorders, maybe we have to pay quite a bit of attention. So just to build on just a little bit what Ian said earlier today. So I think, here we go. Oh, I keep forgetting, I've got two computers here. So what do we know about AMPA receptors? So AMPA receptors were actually the first glutamate receptors where we get full structures of them. So here's the structure that Ian had showed you earlier. They're divided up in 3 different types of domains, just like the NMD receptor. They've got an amino terminal domain, which is involved in the assembly of the AMPA receptor itself. There's a sort of belt portion, which is a ligand binding domain. That's where the neurotransmitter glutamate binds there and actually does all the kind of activation, etc. And then there's also these alpha helical regions, which all come together to form the ion channel pore. And Ian very nicely pointed out this morning that's where you transport different kinds of ions. And the main ions that are really being transported by AMPA receptors are sodium ions, and I'm going to tell you that actually transport calcium ions. If you're not a scientist, then calcium ions might not mean very much to you, but actually calcium ions are actually the signal that triggers a whole bunch of things. And we think that's very, very important in the learning and memory mechanisms that involve in AMPA receptors. So as I said before, people tend to spend a lot of time talking about NMD receptors, and there's a good reason for that, because it's been a long time, 30, 35 years, where people have shown that NMD receptors are very, very strongly tied into neurodevelopment. And of course, now with all the missense mutations, it's obvious why they're linked to neurodevelopmental disorders. So the AMPA receptors have taken a kind of the sideshow, but what's important to remember is that all the learning and memory mechanisms that go on in the brain, so basically the vast majority of all the excitatory signaling that happens in the brain is made by AMPA receptors, not by the NMD receptors. So they're really workhorse, if you like, of the synapse. And just to illustrate a little bit of a conundrum that we started to think about in the lab, I'm going to give you a kind of thought experiment about these. And it's just really, if you start to think about mammals, which are quite diverse group of species, right? So you got an elephant, a mouse, and then a man here. And what's interesting about them is we have different sizes of brain, right? And so we like to think of ourselves as being the smart guys. So we're the guys with all that intellectual capacity. It doesn't mean an elephant, a cat, or a mouse aren't very smart. The elephant's brain is about 5 kilograms. So even though it's got a huge brain compared to ours, this is about 1.5 kilograms, something like that. So quite a bit smaller, but we still, we still pat ourselves on the back and say we're much smarter than everybody else, right? The mouse brain is about 0.4, 0.5 grams. So it's several thousands of fold smaller than the human brain. But here's the problem when we started thinking about AMPA receptors and we only talk about the genes, is that all of these actually have the same size of genome, so they actually have the same amount of DNA. In fact, there's other simpler organisms that have much more DNA than we have, right? But they all have the same AMPA receptors that Ian was talking about, I'm going to be telling you about in a moment. So how on earth is it then that we give ourselves all this credit and say we're the guys with all this intellectual capacity, and then the mouse, as good as it is of stealing cheese from my kitchen, right? It's just not as smart as us, right? It's not a Fortune 100, running a Fortune 100 company out in the west coast of California, right? So what I'm going to tell you about is something that I don't think has been covered in any of these conferences, but it's something that's kind of been always looking in the background and no one's just really tried to look at it. And so what we think, So what we think is that we've uncovered a post-transcriptional program that might not mean much to anybody who isn't a scientist. And this actually very profoundly affects AMPA receptors, affects the NMD receptors, but much, much, much more for AMPA receptors. I'm going to try and tell you why that is the case. What's interesting about it, and this is why it's kind of flew under the radar, if you like, over the years, is that it's taken 30 years to work it out. And it's taken 30 years by different kinds of people. They started off with the cloning of the AMPA receptors. That was the 1990s by Peter Seberg. He was in Heidelberg. And when he cloned those AMPA receptors, the 4 genes that are found in mice, elephants, and also humans, he found that they also underwent these translational processes called RNA editing. I'm going to try and define that a little bit later on. And also something called alternate splicing, right? And that means that just from 4 genes, you can get much, much more types of functionality out of them. So he identified that. So that was in the 90s. And then by the early 2000s or so, David Brett and Roger Nicole, and we were chatting, David Wiley and I were chatting about that last night. They identified these auxiliary proteins which assemble with AMPA receptors I'm going to tell you about in a minute. And so those 2 things are being kind of considered separate from each other, even though they're important. But however, in the recent 4 or 5 years or so, our lab has shown that those 2 mechanisms actually interact with each other. And I'm going to make the argument to you that they actually co-evolved at a very certain time point in evolution. And when did they co-evolve in that certain time point in evolution? It was about 500 million years ago. And I'm sure you all know that that's when, you probably don't know this, but what you realize is that's when the first vertebrates really started to walk about on our planet, right? So what I'm going to tell you about is a very, very ancient mechanism, but the reason I'm bothering to tell you about it is we think that it's actually dysregulated in all neurodevelopmental disorders, including all the GRIN disorders for sure. So just a quick disclosure. So I founded a company called Norse Pharma, and I'm also the chief scientific officer in that. We're actually interested, our focus is really on Fragile X, but we're funded by the GRIN Foundation, so we're actually interested in GRIN disorders. If anybody's interested, we can talk about that later on. Right. So what do we know about glutamatergic synapses? But what we know about glutamatergic synapses really comes from work that was done 30 years ago when people were cloning things. So the early morphologists had this very beautiful image of glutamatergic synapses. And if you were a morphologist at the time, you could tell what was a glutamatergic synapses because they were asymmetrical. So here's the presynaptic side and all these little bubbles here, all vesicles full of glutamate are just about to be released. And on this side here, you can't see it as clear as I can see on my image here, but it's called the postsynaptic density. So it's electron dense when you actually look at it under an electron microscope. And the reason it's electron dense is because it's packed full of all these proteins you've been hearing about in the last day or so, ever since we got here. And so I kind of, myself and David Wiley, is a contemporary of mine, he and I sort of cut our teeth in studying a lot of these proteins, and some people were involved in cloning it. And so all of these proteins are all actually tied into neurodevelopmental disorders. But out of that work from the 90s, which we still kind of hold on to right now, is that there's two very important ion channels in glutamatergic synapses. There's the NMD receptor, which we spent basically all of the last day or so chatting about. And of course, they're extremely important. They're blocked by magnesium arrest. If you depolarize the cell gets excited, that magnesium disappears, and they're very, very good at transporting calcium, and that drives a lot of the plasticity mechanisms. So it makes a lot of sense that this community spends a lot of time thinking about missense mutations in NMD receptors. They're very important. But it turns out that NMD receptors can't really do that job unless the NMD receptor, the AMPA receptors, sorry, which are chucked away out here on the periphery because people kind of think of them as not being terribly important. They're important in depolarizing the cell. Ian very elegantly pointed out that they respond very, very rapidly to glutamate within a few milliseconds. In fact, if you really measure accurately, it's hundreds of microseconds, right? So these guys depolarize the cells, and then the NMD receptor does all the important stuff. Now, I'm going to tell you that that just can't be the case. There's just no way that can be the case. And there's a couple of reasons for that, not really coming from my lab, actually, but coming from morphologists. So the first thing is, First thing is there's been a renewed interest in the morphology of glutamate receptor synapses. Now we know that the AMPA receptor is actually right in the middle, right where glutamate's been released. It's come from some of the groups here from Bordeaux. So there's Olivier Nicole, who's here. So some of his colleagues here actually worked out. There's also some groups in Denver. So our view of glutamatergic synapses, really what we're teaching undergraduate students is probably not really true to what's going on. The other thing, of course, is that the functional properties of these AMPA receptors are actually much, much, much more diverse than everybody thinks they are. And the reason for that is not because of the different types of AMPA receptor genes, but it's because the AMPA receptors, the AMPA receptors assemble with a whole barrage of different types of auxiliary proteins. And so here's the main one here, as we've been studying. There's the claudin family of these receptors. So there's the TARPs, GSG1L, and there's also the cornichins, the CCAMs, and the syndigs. And what's very important to remember, because we've been showing all these structures of AMPA receptors, is that they never look like that in the native system, because native AMPA receptors are always co-assembled with these auxiliary subunits. So if you understand what they're doing in disease, then you need to understand them in the context of these auxiliary subunits. So a couple of things that kind of come out of the work from my lab is that we've shown that in the cerebellum, so it's a part of the brain, it's got at the base of the brain, it's involved in a lot of moral learning and things, then they're mainly kind of tied into these TARPs, these TARP auxiliary subunits. Whereas in contrast, in the hippocampus and the cortex, which is involved in learning and memory, a lot of computational stuff, then not only the TARPs involved in that, but also the cornichins and the CCAMs, so the CSHAs are all involved. So it seems that depending on the complexity of that brain function, then it's matched by the complexity of the biochemistry of those AMPA receptor synapses. So we've actually now been able to come up with a whole bunch of rules based on some of the work that we've done and also by biochemists and proteomic work. So the first thing is that most AMPA receptors are GluE2 containing. So that's a very important thing, because everybody thought until very recently that they're all calcium impermeable, and it turns out that's probably not the case. The other thing is that most AMPA receptors assemble by these TARPs. It seems to be like a default. When you make an AMPA receptor, they always get a TARP, right? And that's very important to what I'm going to tell you later on. And there's an exception to this. So we had a paper a couple of years ago showing that GSG1L, which is really like a TARP, it kind of masquerades as a TARP in synapses, and that forms the exception, but there's only 5% of them. They turn up pretty late in brain development, and they're probably important in terms of epilepsy. The other things, I said before, so TARPs and cornichins assemble, and you also get the TARPs and the CCAM. So how have we approached this over the years? So what we've done, so we recognized this about 10, 15 years ago, that the biology of AMPA receptors are much more complicated. So we better start trying to understand that. So what we've done, this kind of summarizes papers we've had over the 10, 15 years or so, is that we basically reverse engineered the AMPA receptor. We've basically taken the AMPA receptor in the lab, And just like my son's doing, because he's a big fan of engineering, he sort of rips apart engines and things like that and then tries to put them back together again. And so we've basically done the same thing with the AMP receptor. And when we've done that, and here's some of the papers here that we published, we've identified a whole bunch of really important sites which are found on the AMP receptor. And the reason that's important for many of you is the missense mutations that Ian was pointing out earlier in the talk. Most of them are all hotspots around all the sites that we've identified. So I want to just tell you about two of the sites, because it turns out these sites are actually really important in the regulation of AMPA receptors. It's the flip-flop site, which sounds very fancy kind of name. And then there's also the QR site. So the flip-flop site is a site for alternate splicing. So that's where, you know, a gene can make different versions of itself. So the flip is one version, flop is a different. And it turns out the flip-flop site interacts with auxiliary subunits that I just told you about, and it can either switch them off or switch them on. And that's a very important thing to think about, because your brain's constantly changing the flip-flop content of it. I'm going to show you that in a minute. The other thing is the QR site. So the QR site's down here. We just recently had the Nature paper talking about that. And the way we used to think about the QR site, it was basically an on-off switch for calcium. So if it was a GLUE2 containing, which is most AMPA receptors in the brain, then those AMPA receptors were calcium impermeable. But it turns out when you start to recapitulate the AMPA receptors in the brain that have these different auxiliary subunits, then suddenly they start transporting calcium. So there's a much greater continuum of expression of AMPA receptors with calcium permeability in the brain. So just to kind of quickly zip you through some of the things that we found over the years. So in terms of the TARPs and how they work, so we know for a long time, and this is work that came out of our lab, but other people have shown something like this for 30 years. You can see the different cell types, the hippocampus cerebellum, for example, have different kinetic properties. And that's because these AMPA receptors, it's not because of their subunit composition, which people always talked about. It's really because of the different, it's because of the different TARPs they express. So here's one type of TARP, another one, and GS1L, and you can see that all the properties change very much of these same AMPA receptors depending on the auxiliary subunits that you find, right? So from that, there was a bunch of rules, and I won't take you through them. You can go and dig up our papers on this, but the TARP isoform is important. The stoichiometry, whether you have 2 or 4 TARPs onto them, and also the flip-flop cassette, and I'm going to come to that in a minute. So the flip-flop cassette, so this comes out of a neuron paper, 2 neuron papers we had over the last few years. Flip-flop cassette, it turns out we identified that it binds to the base of the ligand binding domain of AMPA receptors, and it can interact with it. And if you have a flip version of the AMPA receptor, then it engages with the TARP, and you get that prolonged synaptic event. So these very prolonged synaptic events that people see in hippocampus is because they're flip dominant. However, even if the TARP's present, but you've got a flop version, then the TARP doesn't affect the AMPA receptor. And the reason that's important is you can get a whole variation of different kinds of signaling properties of AMPA receptors and synapses, something that really hadn't been appreciated before, right? So, yeah. So why is that important? I'm going to jump over the RG editing of the RNA edit in the RG site. Why that's important is if you look at the developing brains, this is the rodent brain, everything starts off as flip. So all the TARPs would be switched on at that time. But the problem is that the TARPs don't really express until you get into the adult, right? So it's not really taking advantage of it, right? The flop, which switches off the TARPs, actually slowly develops during development, just as the TARPs kick on. So there's a kind of yin-yang thing going on with the TARPs. Are the obvious, the flip version means the TARP works, but there's not many of these clons around at that time. So as the flip and flop come in, and they're about 50% together by the end you get into the adult, then it means you've got this great way in which to regulate very, very finely the kinetics of the AMPA receptors and their signaling properties. So I think it shows you that there. So I'm just going to switch now to something in the last 5 to 8 minutes or so. And it's kind of went back to that. We said, well, if that's really true for alternate splicing, what about RNA editing of the QR site? So for 30 years or so, up until just recently with our paper, the way people tended to think about AMPA receptors is that there's GluE2 containing, and these GluE2 containing AMPA receptors are calcium impermeable. Because this is the very sciencey audience, then I'm not going to tell you how we did that, but we can measure calcium permeability. Here's the experiment there, and you can measure it. And what you find is because of the reversible potential of these experiments, they're not very calcium permeable. So that's how people kind of used to think about things. Most of our neurons are thought to be like that. And then there's some cells, this is actually a Bergman glia, which is not a nerve cell, it's an astrocyte, the cerebellum, then they're very calcium permeable. But that's because they do not have the GluE2 subunit. But we said, well, hold on, we've just learned all this stuff about the association of AMPA receptors with auxiliary subunits. Maybe things change when you actually try to recapitulate the AMPA receptor that's in the brain rather than just creating a recombinant receptor that doesn't have any of these auxiliary subunits. So we started off by looking in the cerebellum. We had previously shown in a neuron paper 4 or 5 years ago, we looked at stellate cells and also Purkinje cells. Here's the AMPA receptors found in stellate cells. They have 2 TARPs. And then we looked at Purkinje cells. They've got 4 TARPs. And it turns out if you recapitulate them in the lab, this is what we found here. So here's an AMPA receptor that literally does not exist in your brain, right? There's no TARPs on them. But what we did is it engineered the TARPs so you'd have 4 TARPs. You might think that would give a huge a boost on calcium permeability, but it didn't. It was only when we put the TARPs just on the GluE2 subunit, so it's an asymmetry to this, then suddenly you start to get an increase in the calcium permeability of these AMPA receptors. So AMPA receptors are absolutely involved in calcium transport in central synapses, but on a very fast time scale. So what about the hippocampus and the cortex? Biochemistry is much more complicated there because not only do you have the TARPs, which are found here, okay, but you also have these cornichons, so they're octamers, so they're really boosting up the maximum, kind of biochemistry you can have here. I'm not going to show you the data in the interest of time, but what we found is that they even boosted the calcium permeability even more, very dramatically, in fact. It even surprised us in the lab. And so we often did a whole slew of these, right? We literally did a whole bunch of these. And what we found, so I'm going to draw your attention to this part of the slide here. So I'm going to talk about this in a second. But what we found is rather than having GluE2 containing AMPA receptors being stuck down here, not being very calcium permeable, and then the GluE2 lacking ones being up here, like the Bergman glia, so you just have 2 populations, what we found is there was, in fact, an entire continuum of calcium permeability of AMPA receptors that we find in the brain. Makes sense, right? If you want to have a very dynamic brain, then you want to be able to pick and choose the calcium permeability you want and the signal you get. It seems very obvious, but for 30 years, we didn't think that that was the case, okay? Now, what's interesting for people here who are interested in neurodevelopmental disorders, we went in and actually looked at 3 kids actually, that had missense mutations in the QR site. We'd also identified this site G site, which one of the researchers was chatting about from Bologna was chatting about yesterday. We also looked at this +4 site, and what we found is that when we looked at the missense mutations here, then rather than being in this kind of rather controlled calcium permeability, then the calcium permeability got much, much greater. And so what we think in these kids, the reason they have such detrimental effects on their brain function is because the calcium permeability is just too high. Right? You should have enough calcium permeability, but once you get beyond too much of it, then it starts to become detrimental for these kids. However, there's another kid who also has all these features. And what we found with that is actually there was a reduction in transport of calcium. So it means there's a certain sweet spot you need to have in your brain. Too much is bad and too little is bad as well. So it means that there's something really to be understood there. The reason that's a great framework to start to think about AMPA receptors in the brain is that if you then look at, and this is, we've just taken some of the missense mutations that we've had from other people. Here's the AMPA receptor pore structure that we identified, site G and QR site. And if you look at the 4 different types of genes that are in AMPA receptors, you can see there's a whole bunch of missense mutations that are found there that we think will all affect calcium permeability in different ways, up, down, or change the gating and the calcium permeability all at the same time. So when we published our paper in Nature, a bunch of people wrote to me and said, oh, great observation, But it's really surprising. How did nobody see that before? And it turns out people did see it before. So if you go back and dig up Peter Jonas's paper from 1995, and I know Peter very well, and so he's a great scientist, but what you can see is that he actually found a whole range in calcium permeability. It's just what happened at the time. We didn't have these auxiliary subunits, so we just dogmatized things and said, there's calcium impermeable ones, and there's calcium permeable ones. But it turns out we always had that evidence that there was a continuum of these. We've added that as well. There's another piece of evidence to suggest why that would be the case as well, because if you actually get, this is just to tell me I've got 5 minutes left. It's usually my wife that's calling me to pick her up or something. But anyway, sorry about the noise. So if you go back and look at the ancestral forms of AMPA receptors, it turns out during evolution, so it's only actually very recently with the mammalian brain, that we've actually controlled the amount of calcium. But if you go back and look at very simple organisms, so here's the parts of the protein of AMPA receptors that are important in calcium transport. So here's site G. I know this is a bit crazy for people who are not used to looking at science. Here's the QR site, which can be unedited in GLUE2, edited in GLUE2 and not with the other ones. But if you go back and look at Aplysia, which is a sort of sea slug, if you like, that a lot of neuroscientists study, you can see that all of their AMPA receptors are unedited. So they're calcium permeable, right? And if you go back to trichloplax, so probably many of you don't know what it is, but it's basically a carpet of cells which is found in the CNS, and it's a favorite organism for geneticists. And all of them also have unedited AMPA receptors. In fact, there's no RNA editing there. So the default in evolution was to have glutamate receptors, which were calcium permeable. It's only as our brain became sophisticated that we had to control that calcium permeability, but we never shut it down fully. And I guess that's the important thing. So on that evolutionary field, if you go back and then track and do some phylogenetic analysis and look at the genome structure of AMPA receptors, what you find is that the AMPA receptor subunits, not only we found in the vertebrates with nervous systems, you go all the way back to here, and in fact to the trichloplax, which is one of the simplest organisms, multicellular organisms we know. They actually a whole bunch of different types of AMPA receptor genes. So the AMPA receptor genes that are in our brains, they actually were hanging around 700-800 million years ago. The diversification of AMPA receptor genes is a long, it's a very, very ancient thing, happened in very simple organisms. But in terms of alternate splicing, which I've been talking about, and RNA editing, then actually that happens only when we started to form vertebrates. So it was the occurrence of vertebrates going from very simple vertebrates to more complicated ones that we got alternate splicing and RNA editing. And that coincided, and I don't think it's a coincidence, it coincided with the expansion of the different types of auxiliary subunits here. Right. And if you're interested at the end, I'll tell you reasons why we think these must have co-evolved with each other. So I think it's a really exciting time to use this as a framework to think about CNS disease, right? What we're looking at is something which is very, very ancient, starting with the beginning of vertebrates, right? So we've had this all around, right? But it's obviously been optimized in the human brain. And the key thing is to understand how does that play its role in the CNS disease that we're all very hopeful in trying to kind of find some cure and things like that. So this is my last slide, just to kind of finish on things. Just finish on things. I just want to kind of put it into a time concept for the scientists here, because many have lived through this. So we're really moving from chapter 2 to chapter 3 of the glutamate receptor story. So way back in the early 90s, when I was starting my PhD, and some of us in the room were doing the same thing, then a number of labs, but one of the really notable ones was Peter Seberg. He actually cloned the AMPA receptor genes at that time. And then he was a very smart guy, very astute guy, and noticed that they had this alternate splicing and also RNA editing. So that was the first indication of it. And he couldn't have known how complicated things were going to be. It was already very amazing finding at the time. But then what's happened over the years, so with David Brett, Roger Nicole, and then a bunch of other people, so I've been working a lot with Bernd Fackler in Freiburg, who's a fantastic proteomic expert biochemist, and they've been working about how you get the assembly of these different kinds of AMPA receptor subunits. And our contribution has really been saying that the occurrence of these actually affects the properties, alternate splicing and RNA editing, something that hadn't really been appreciated before. So we're getting towards the end of the second chapter, if you like, of the glutamate receptor story. And this is where we can really start to have an impact as we start to think about disease and how we can fix it. But where are we heading now? So where we already are, because our lab's already kind of moving into this area, is why do you have all these auxiliary subunits in the first place? So one of them is because this very important regulation of the signaling of AMPA receptors. But the other thing we're finding is that when they get these different jackets on of different auxiliary subunits, then that puts them into different kinds of synapses within even individual cells. And so the interesting thing is going to be in the future is what happens with kids with GRIN read disorders, et cetera. Is that connection just on the individual synapses? Is that something that we need to be thinking about to correct? Right? So just to finish, just want to thank all the people that's been involved in this work, so all these people in my lab who's been doing this over the year, and our collaborators, and there's many more from this, but these are the ones who we're working with kind of on a regular basis. And I just want to thank Fede, who's from Barcelona, actually, and Shintong. There are two postdocs in my lab who are involved in that Nature paper. And I also want to thank GRIN for funding some of this work and having faith that we can actually pull off something interesting. We're also interested in SYNGAP because we actually think it's very, very similar. We think the problem is really the same problem there. And then funding, within Canada, and here's the other people in the lab. And for your attention, so at the Q&A session, I'll be happy to chat more about this. So thanks again for your time.